I'm going to go ahead and get started as more people join us, just with an introduction of this series, and then I'll introduce our speaker today. Today's session is part of a multi-year series that is presented by our Global and Local Center for Mental Health Disparities. As you guys know, I'm Maria Prom and direct the Global and Local Center, and this has been a really wonderful opportunity we've had over the last few years to invite international scholars and researchers to join us, either virtually in more recent times, but previously we've also had the opportunity for visitors. This is the Global Minds and Local Insights Mental Health Research Scholar Series. This series is sponsored through the donor fund that was developed by Peter H. Knapp's family in order to support lectures devoted to research in the field of psychiatry to Boston University School of Medicine and the Department of Psychiatry. Today's speaker, we're really excited to announce a very important and relevant topic to us here at Boston Medical Center. We will have Dr. Liam Mutesa, who is joining us from Rwanda this morning. And I'm going to share, now that we've got a few more people in the room, I'll share a little bit about him as he pulls up his slides. Great. So Professor Mutesa, MD, PhD, serves as the director of the Center for Human Genetics at the College of Medicine and Health Services at the University of Rwanda, a center he established in 2006. He obtained his MD from the University of Rwanda in 2003 and earned his PhD in 2009 from the University of Liège in Belgium, where he also completed his postdoc in 2011. He's had significant involvement in academics, clinical genetic research, genomics, epigenomics, as well as building research capacity, and they've all been hallmarks of his very successful career. He's held several prestigious positions in the health sector in Rwanda and is a pioneer in the field of genetics and genomics. He spearheaded the development and implementation of Rwanda's Reference Center for Human Genetics, the only facility of its kind in East Africa. And he's the PI of several research grants funded by NIH, EU, EDCTP, NIHR, and the Wellcome Trust, among many others. He's a research pioneer of transgenerational epigenomics of trauma and PTSD in Rwanda, a research program that focuses on the transgenerational transmission of the epigenetic impact of the genocide exposure and PTSD in women survivors and their offspring. He supervised over 100 trainees from master's to postdoctoral fellows in both Rwanda and overseas. With more than 230 peer-reviewed publications to his name, he's a prolific contributor to research and in the scientific field. His outstanding contributions has led to him being recognized with the prestigious HUGO African Prize in 2024, and he's a fellow of the African Academy of Sciences. I'm delighted to introduce Dr. Mutesa and really excited to hear about his work that, again, is very relevant to our population here at Boston Medical Center. Thank you, Dr. Mutesa.
Thank you very much, Dr. Prom, and I'm happy just to be invited to speak here and then share the experience of our study from Rwanda, which just focus on intergenerational transmission of PTSD, post-traumatic stress disorder. Actually, this is just a more than 10-year project that we implemented here in Rwanda as a consequence of the 1994 genocide against the Tutsi that happened here in Rwanda. Actually, just to zoom in to show you, this is just the Rwanda, the small red spot you see here on the African continent. So with a high density and with impressive outcome after 1994 genocide and improvement. You can see the life expectancy is reaching 70 years, and the GDP per capita more than 1,000 per here in Rwanda. So it's really among the few countries that are having a very impressive dynamic economy during the last 3 decades. So this country has a very dark page. The genocide that we just what happened in 1994 actually did not start in 1994. 1994. It started in 1959 when the Rwanda was transitioning from the colonial period to independence and also republic system. So then the ethnic, the Tutsi ethnic people were just starting to have conflict with the Hutu ethnic in this country due to the different ideology that were just being maintained here in these countries. And then in 1994, this is really the tragedy. More than 1 million of Tutsi were killed, and you can see the country how it was really heavily destroyed. And this really resulted in the consequences of the survivors with different problems. And among these problems, we have today, we are dealing with the PTSD consequences. So I was born in India, grew up in this country, and actually my curiosity as a scientist starts really very young when I was really trying to do some raising. I was raising some rabbits at my family house, and that time I was really interested by the growing reproduction. And then I was not really effect of a consanguinity. You can see this was my population. So genetically, you can see the high reproductivity of my population, so which was very impressive for me. And I was not really unknown what could be the consequences of the consanguinity, which happens actually. I could see some of the rabbits with severe congenital malformations, some with albinism, agenesis, and the other malformations, a lack of different organs. And then this was just the factor that I was not really aware about the law of segregation. So I come just to understand this after my studies and also after doing some of the genetic research projects. And then I published this story in 2022 just to share with the scientific community. So some of the studies I did actually when I started my postgraduate, my PhD especially, were just related to the problems that I wanted to answer here in this country. For example, the malnutrition was a problem after 1994 genocide. So the disease called cystic fibrosis is more common in the Caucasian population, but some of the patients that I had in Rwanda were expressing some symptoms that could really suggest an autosomal recessive condition. And then I started really working on this disease to understand if this could be the case in Rwandan population. So we discovered new genes that were modifiers genes, like these ENAC genes and the CFTR genes, which is the cause of this. And then I started really understanding some of the concepts that I could not really understand when I was in the childhood period. And then when I completed my studies in 2009 to 2011, so I came back to Rwanda, but I have started just the work of creating the first Center for Human Genetics here in East Africa in 2006. And then the major challenge that I really faced in this period after 1994 genocide, and then when I also completed my studies, was just to see that the genetic was not really a priority, which means I could not really get support from the different structure, the different public institutions and our government institution, because the priority was not the genetic, rather than other problems like infectious disease, HIV, and others. So one of the strategies that I really implemented was really to raise funds from the grants. So I started just writing grants. And you can see different funding institutions that way I have been submitting my grants since, for example, 2010. So this is just the ratio of the grants. We have been just with my team have been very active, just writing grants, but not really being successful 100%, but at least to 24% during the last 15 years almost. So, but it's a huge work, really. We produced, and after this work, sometimes we got rejected, but those few grants we got really were really very powerful, just useful, just to start our addressing our works. And then that's why one of the projects that really was very interesting for me was this intergenerational transmission of post-traumatic stress disorder to understand the consequences of a 1994 genocide. And then, of course, there are different traumatic events that will lead to PTSD. You see here different genocide in Armenian population, Holocaust, of course, our case. Now what's happening in Ukraine, Gaza, Israel, the war here in Sudan, and also in our neighboring countries. So all of these issues, these traumatic events, they lead to post-traumatic stress disorder, which is being expressed by different symptoms like avoidance, nerves, depression, anxiety, and so on. This happens really in our society after 1994. You can see, for example, on this slide, some of the studies done here by my colleagues, where they try to compare the PTSD in the general population. You can see in the genocide survivors still high, 28%, while it represent around 4% in the general population. And also depression very high, 10%. Comparing to the general population. So, and then sometimes this case can lead to some chronic disease and suicide. Something which is very interesting is just to see the genetic diversity of Africa, in Africa, of African population. But when you just zoom in a epigenetic study, the landscape is still really unexplored. So we did some study to understand the African genetic diversity and adaptation, which could just inform the precision medicine. And then this is really one of the gaps that we identified. Even though some studies done before to trying just to correlate the epigenetic transmission of PTSD were really predicting this effect. For example, Professor Yehuda, she has been doing a lot of studies in the Holocaust population. And then trauma survivors. The same prediction was confirmed with our studies now 3 years ago, after doing this intergenerational transmission PTSD and the epigenetic study that we did here, that I'm just going to show you in a second. So, we know very well that the epigenetic mechanisms are affected by different factors like development in utero, environmental factors, aging, diet, but also some health conditions like cancer, autoimmune disease, and the mental disorders can just affect this. But what we did not know up to now was just the trauma caused by the epigenetic caused by the war, PTSD, but also intimate partner violence, among others. Of course, these were also supported by different studies in animal models, which showed just the stress, the mice which were submitted to stress, developing a lot of PTSD symptoms, but also symptoms like obesity when it compared to the weight type. So we try just to understand this in humans using our case in Rwanda, but also trying to understand the concept of transmission, intergenerational, multigenerational, and transgenerational. So we have been focusing actually in our study in this intergeneration for the 2 generations. Today, we are also looking into the 3rd generations. So this is how it is going to be reflected over generation with these evidences that we are really displaying in our studies. And of course, some studies also have predicted the consequences during in utero when the mother just is exposed to some kind of different kind of type of trauma, then this can lead really to elevated risk of mental illness later in the life of the children. So this was really the case. These are our children who were born after 1994. Genocide, full of dreams, as you can see here, to be doctors, teachers, pastors. So really, this was very impressive to see these children born, they have this. However, in this country, since 1994 genocide, we celebrate, we commemorate. We do our commemoration, which just coincide with April, July, which is the period of the genocide. Actually, the genocide was carried out within 100 days. So what we have been just observing, these children we see here, they were trying to express different symptoms related to PTSD, like depression, anxiety, hallucinations, among others, but especially the poor performance in schools. So we wanted to understand what's happening, and that's how really our initial research question started with this observation. So this started a little long ago, almost 20 years ago, when we were really seeing this happening. And then in this country, we really focused on one of the organizations called AVEGA, which is actually the Association of Genocide Widows, women who were trauma survivors, like this woman you see here. So, and then the approach we used in our first pilot study was trying just to, of course, to understand their lived experience at that time, but mainly focusing on the PTSD symptoms evaluation, but also methylation studies. Of course, for the PTSD, we used the Africa tool. We worked with different psychologists because this did not only support the data collection, but also understanding of the PTSD, but also provide some psychotherapy for them, working with different clinical psychology facilities to provide support to these women. And then what we did as a pilot study in 2012, we collected samples from this case, the mothers and their children who were living in Rwanda during the 1994 genocide, and our control case were actually the mothers that were living outside Rwanda during the 1994 genocide who were also pregnant, like the mothers who were in Rwanda during 1994 genocide. So this was only the way we could get our control, very sharing some characteristics. Even though they were living outside Rwanda, because as I mentioned, the problem of genocide started in 1959 with different conflicts happening, and then this population, this Rwandan, they got a chance actually to leave their country, and they have been refugees. Otherwise, they could have been also a victim of the genocide. So those were really our control. And then our first result from this pilot were really impressive when you compare, for example, the mothers and the children, both controlled and exposed. You can see here in the green, those are the controls for mothers and children, but there's a high level of PTSD and the BTI in exposed mothers and their children. And very interesting also, when you look into the HPA axis, just to understand the level of different biomarkers like cortisol, you know very well that this HPA axis and also the negative feedback when there is a stress, the hypothalamus will just stimulate anterior pituitary and then adrenal cortex, different hormones will be produced. And then we wanted just to see what is the level of GR and the MR and the cortisol. So you can see the level of cortisol was a bit low when you compare to the controls from this case. The same here in black, the level of GR, glucocorticoid, mineralocorticoid for the cases was they were expressing different variations when you compare to the control, our controls. So these were really our first evidence in this context, but we went far to study some of the genes that is involved in methylation, like NR3C1, in these two population. And then you can see again, the level of CPG was very high. There was very high methylation always in exposed children and mothers who were really exposed to genocide here in Rwanda. So our first evidences really prompt us just to expand our study on country level. And then we got NIH grant in 2017. We expanded a little bit our sample size. You can see we used the same approach, but we used, in this case, we did epigenome-wide association studies by methylation study, doing a methylation study using Illumina chip. This Epigenetic chip that could just allow us just to scan the large epigenome level to see different methylations. So in this epic chip, so the first aim was just to focus on glucocorticoid receptor regulator network genes, where you have a rich genes, more than 82 genes being identified in this context. And of course, so yeah, this is more technical. So of course, doing this, we could see our sample was subject to different controls and also quality check. And very interesting, even we could do the sex discordance. For example, here you could see the Y chromosomes for those children born in 1994, just to our differentiation between sex. So the most interesting here are these results you see here on this slide. So we identified 3 genes: BCOR, PRDM8, VWDE. Those were the genes highly methylated. In this case of genocide exposure, you can see the pink zones. Those are really the high methylated zones. Actually, the methylation level is just evaluated from 0 to 1. The more you are close to 1, the more you get the high methylation. The more you are just close to 0, that means that the methylation is very low. And then you can still see in our exposed group versus controls, you see the high level of methylation for the 3 genes. Again, if you take one gene from the 3, for example, this VWDE, you can still see the high methylations in mothers and the children, both exposed to 1994 genocide when you compare to the controls. And very interestingly, in this study, we also identified several DMRs, differentially methylated regions in exposed mothers, for example, versus non-exposed mothers. You can see the different genes. The same for children. Several DMRs were really identified as this was really a very good evidence when you just have a combination of both groups. 6 genes were showing a very high level of methylation, DMR, and these genes are correlated with the brain and and the symptoms that we have been seeing in our population. Related to anxiety, depression, and then other mental health issues that could just be explained by this. And these also genes are also very good target for precision medicine. So we also went far to understand the implication of HPA axis mechanism and also other biomarkers. Of course, we've more focused on this, but also other biomarkers. The biomarkers we added were the BDNF, which is a brain-derived neurotrophic trophic factor, which is more expressed in the central nervous system and linked to several mental health conditions, especially to the low level of this can just lead to different problems like bipolar disorder, schizophrenia, of course, PTSD, and major depressive disorder. So this was one of our targets in these biomarkers. Another biomarker we tested was a neuropeptide, which is a signaling molecule in the brain that can affect the mental health by influencing neurotransmitters in the brain, but also related to PTSD and the depression and anxiety. Another one was the PACAP, this pituitary adenylate secrase activating polypeptide, which is also expressed especially when you have high stress in an individual. And the last one was the HSP, the heat shock protein. So we also wanted to explore the level of this in our population, which is also the increase of this HSP has been just reported to be associated with anxiety, depression, and the PTSD. Of course, in this study, we tried just to correlate our PTSD symptoms in the three groups. The first is just the generation one of mothers, and then generation two of first child, the child who was born in 1994, and the child who was born after 1994, the young sibling. So you can see here in red the case, expressing high symptoms of PTSD when you compare here in blue with the control. So different parameters were just measured here, exposure to trauma, still see our case very high. PTSD symptoms still very high in mothers, child 1, child 2. Same for intrusion, PTSD intrusion, avoidance, negative mood, negative cognition, and resilience, resilience, which is very good. Just to see there is a slight decrease of in this psycho resilience in this group for mothers and the child 1, and so on. And also, in addition to these symptoms, we try to correlate, of course, the cortisol. You see, again, the correlation, the variation, glucocorticoid, mineralocorticoid, like in our pyruvate. The HSP 27 were also very high here in the case versus control. Same for PACAP variation, especially in the first child and the second child. Slightly low in the mother, and then BDNF very high and the low neuropeptide. So those variations were really put together to make kind of combination to understand very well the intergenerational transmission of these biological markers. And then when we did the correlation, some genes that you see here that at the genes that from the DMR that we identified, we try to correlate in the mothers, for example, here with the HPA markers. You can see here in red, some genes we expressing a very high correlation. Here, for example, with this correlation test, the same, this is just Pearson test, but the same here with this other test, correlation test, still the genes with high correlation with this glucocorticoid and versus the DMR. This was a very good evidence. So you can see again still this gene, which is expressed here, expressing a very high correlation on this figure. And last year, of course, I'm sure you have seen this study from Syrian refugees where they tried just to see the epigenetic to identify some epigenetic signatures over generation, 3 generations actually. You can see these 3 generations with several epigenetic signatures that have been identified. So our current study was, has been awarded by Wellcome Trust, is trying also to understand the intergenerational transmission across generation, and trying to see if we can identify more epigenetic signatures in our population, like they did here in these Syrian refugee populations. Of course, this study have been really raising international public attention. You can see even in Gazan's children. This was an article that was published last year, just showing how this is just a reflection of what is going to happen, similar to what's happened here in Rwanda for the intergenerational transmission of post-traumatic stress disorder. Then, of course, I may say that Rwanda, we have been lucky to have some parameters to address the resilience during the last more than 30, the last 3 decades, I may say. Even though the resilience is a very big complex, but our particular case here, for example, some of the interventions that happened in this country, or strategies really were paying back, and also contributing to resilience. So it's that with reconciliation of the population, and also building the country. You can see this country is having a rapid socio-economic and the security transformation. You can see the improvement of infrastructure, tourism. This is really happening here. Last year, for example, we hosted this UCI championship in Rwanda. This is just some of the examples. These are some of the examples of the resilience. Even the education has been improved, the political, the women empowerment, the political and the security stability and the leadership. So this is the country today. This is really a sign of the resilience, which also contributes to the decrease of symptoms. But however, we still have to explore, and also to make sure that we try to correlate with this resilience with evidence, biological evidence. So those are the study we are doing. Our current study, which is funded by Wellcome Trust, has 2 aims, actually. We are focusing on the transgenerational transmission of PTSD and the epigenetics, but also the lived experience. In this study, we're trying to have this mechanistic insight, which is really a quasi-experimental design, to understand the causal factors. And from this mother who were pregnant, these are just population. So the exposed and unexposed, this is just the total sample size we're targeting. And again, we are doing the methylation, DNA methylation studies, and the HPA axis, the other biomarkers evaluation. But the most important is just the evaluation of the resilience by a lived experience. So our lived experience actually built upon this community. So on above here, these are just the women who are participants in our studies. This was during the community engagement in our second project conducted by our PhD student who graduated, Dr. Musanabaganwa. And then also we have the Ibuka, which is NGO with the mission to support genocide survivors and the programs devoted to peace building in a country. So the 2 organizations are very key in this in these studies. So they are part of the study. They are also having this kind of ownership. That's how they express, and then we can just work together to address some of the aspect of the studies, including, especially currently, including the lived experience. So I just want to finish by showing you some of the current preliminary data. So this is our this is our country. So you see on these green zones, that's where we try to collect samples across the country to understand the genocide consequences with different geographical locations. So we are recruiting our participants across the country. So we are also using a new approach. We are trying to use the pedigrees, having the 3 generations. So the generation one, of course, the mothers here in the black, the mother who was survivor, trauma survivor, and their husband, of course, here you see they have been killed, and then the children from this family. Those in the black, those are the individuals who are being recruited in our study. So here I want just to show one case, which is very surprising and was impressive. So I want to show you this family 6 with 3 generations again, you see. So this was a testimony of the high PTSD symptom across generation. You see in the middle here, the mother who was trauma survivor in 1994 genocide. She lost her husband here. The husband, H1 you see here, who was killed. And then this mother was not legally married with the husband one, and then they got both of them, they got 3 children, you see in generation 2. This child 1 and child 2 were killed with the father. And then the child 3, this is the one who was born in 1994. And then the mother also got married. Actually, he was not legally married with the husband too. And then they got 2 children. You see child 4 here and the child 5. And then what you see, the observation that we see today, for example, when it starts from the mother, so the story from this particular case, the in-laws, actually the in-laws of the husband one had intrafamilial conflict with the mother here, with different disputes related to asset like lands and so on. And then they chased out the mother, and then she became homeless and extremely poor. And then this linked to high PTSD symptom to this mother. Not only this, even when you go to generation 2, the descendant of the children from this mother, you see the mother has 5 children. The particular case that we are seeing here, which is common to other families that we are recruiting, majority are unemployed, and some are being engaged in drug abuse or prostitution. So the particular case here of child 4 here was raped, for example, here by several multiple men, and then the father of this child, grandchild of the mother, is not known. And even the mother was left by this, in this situation, she contracted actually HIV, and later she developed PTSD and the suicide ideas. You see in generation 2, it's happening. And later on, she met another man here, the husband too, you see here, and they got pregnant, and again had a child. This is a grandchild of this mother. And then, unfortunately, this husband also left her with a lot of problems related to financial and emotional struggles. So when you also come back here, so this mother who's a trauma survivor of 1994 genocide has been also under pressure with the second husband, because the second husband has just hiding relationship with another woman, and it seems they have had several children together. So you understand, even if you go even in generation 3, the grandchildren, they become really responsibility to the mother who was a survivor of 1994 genocide. This is the reality. This is the real case of what's happening today. So this is just to show you the current study we are conducting to trying to understand the lived experience of the PTSD and also intergenerational transmission. So to finish, the whole study actually has showed the proof of concept of intergenerational transmission of PTSD symptoms among trauma survivors, the genocide exposed trauma survivors. And the use of EWAS in our project 2 was very useful, which allowed us to identify several CPG sites which are correlated with mental health conditions that are subject actually to precision medicine. And these are also some of the aspects in our current studies. And the HPA axis correlation shows also biological variation. You have seen several variations in the HPA biomarkers and other biomarkers like neuropeptide, PACAP, and the HSP 70 and the 27. And the final current focus and current target, just trying to understand what's happening across generation, especially with more focus on the lived experience, but also trying to correlate this with the resilience, which is a very positive, has had very positive impact in this country, but we think that this has also contributing to the decrease of symptoms in this population. So I just want to thank my strong collaborators, especially Monica, Professor Udin, and Professor Weidman from the University of South Florida, with whom we started this project during the phase 2, the project 2, and other colleagues, of course, from different universities, USF, of course, Shegun from Queen Mary, University of London, Rose Jimini from VUB in Brussels, other colleagues from Netherlands who supported the studies for the cognitive aspect, and of course my colleagues from the University of Rwanda, psychologists and geneticists, but also the two organizations, Avega and Ibuka, who are our main partners in this. Of course, we have benefits from different funding institutions, support from Wellcome Trust, NIH, through H3 Africa. Thank you very much. So that was my talk. Just I'm happy to take any question.